Good evening. My name is Johanna Koljonen and you are watching Crosstalks, a collaboration between two of Sweden's leading universities, Stockholm University and KTH, the Royal Institute of Technology. Please do join us in discussing today's topics on Twitter, where our handle is CrosstalksTV and our hashtag is Crosstalks. What do we really know about the human brain? More than ever, but not enough by far. In what could be described as our generation's race for the moon, major international research initiatives are setting out to map the physiology and functionality of the brain. Here to discuss some of the promising paths towards understanding this, our most complex organ, are Anders Landsner, professor of computational biology at Stockholm University, also affiliated professor of computer science at KTH Royal Institute of Technology, Marie Öhman, professor in molecular biosciences at Stockholm University, and Håkan Fischer, professor in biological psychology at the Department of Psychology at Stockholm University. Welcome to Crosstalks.
Thank you.
Thank you.
So, Anders, you're a computer scientist using ICT tools to understand biology. How did you become interested in the human brain?
Okay, that's a story starting long ago, in the mid-60s when I was 17 years about. And everyone was interested in politics and building a better society. And my take on that became, to build a better society, we need to know what people like. And to know what people like, we have to somehow look into their brains. That was my conclusion at that time. So I started to read about the brain with that purpose. And then I understood that after a while, no one knows this. It's a mystery. And
some people would have responded to this by growing their hair long and dancing in flowered clothes, but you made another choice.
Yeah. So I got fascinated, and after some time, I heard people started simulating neurons with computers, and then I went in that direction.
You're currently working with the European Human Brain Project, a project that launched with the goal of being able to simulate the structure and function of the human brain in a computer. Is that what it sounds like? Are you trying to simulate, to build essentially a working copy of a human brain, or just a very good model, if there's a distinction there?
Yes, we are. Yes, I mean, the vision is to build a model, a working model of the brain. And doing it in 10 years, of course, is a great challenge and maybe not altogether possible. But the real core of that project, I think, is to bring more of IT, more of information technology, computer science, and such technology into brain science, because and especially the component where you model the system, build models, network models, and you simulate them on supercomputers, for instance. Because that is a way to help bringing together all the knowledge and data we have into a model we can understand. That's the...
But I think this may be a completely foolish layman's question, and then you're right to tell me so, but I'm not sure about are you trying to take all the existing knowledge about the brain and make all of those models work at the same time? Or are you trying to make a synthetic brain and look what it does? Maybe the answer is both.
Well, yeah, I mean, we are trying to maybe first build a simplified model of the brain, because the brain is extremely complex. We can't certainly not use all the detail there that exists. And then we can, as a strategy, detail certain parts of it and look more in depth on those. And different research groups can use this model and detail it in different aspects. And in that way, we want to contribute to the understanding by bringing things together, by understanding how cognitive function can relate or is implemented at the neurons and synapses and the dynamic activity in those levels. There is maybe no other technique you can use to do that, actually. So this synthetic model building is very important.
That's incredibly exciting. And this, so is that specific relationship what you're trying to learn with this approach, or are there multiple goals?
Well, yeah, there are multiple goals. Understanding is, I think, the key thing. But once we understand better the brain, we can, of course, understand diseases better. We can hopefully help in drug development in the future with good models. And the other aspect is we can take these functional models and implement them maybe on chips and neurochips, and then use those in different applications, technical applications. So that's another goal of the project.
Human brain functions in machines, essentially.
Yes, in some of it, and in the future, yes.
Exciting. Marie, you work with the smallest parts of the brain, so specifically looking at enzymes that affect the RNA molecules in the cells of the brain. Can you explain how you do that and what that tells us?
Yeah, so my research started with the biochemistry of an enzyme. So this enzyme is changing the genetic code, but not in the DNA. It's in the RNA, and the RNA is a replica of the DNA, and it's a template for proteins. So as you can see here, you have and this is something that is changing during development. So what you can see here on this picture is a set of cells. It's brain cells from a mouse embryo. You take them at embryonic day 16, and then they can grow on a plate. And in the beginning, these cells don't divide, but they start talking to each other. So they build connections and synapses, and after 14 days, you have a developed network between the cells. So the blue parts here, they are the nucleus of the cells, and then all the connections is the dendrite connections and axons and synapses.
So this is a picture of a developing brain,
yes,
essentially, not in an embryo, but as it would be inside an embryo.
Yes, so this is our model system to study this enzyme. So what this enzyme is doing is that it's changing the RNA that is the code or the template for making proteins. So if you look at the beginning there, on day 1, you have a TAG, and that TAG is changing. So later on, it's a TGG. So that means that the code is changing. So you have a different amino acid code. So this triplet is coding for an amino acid. So in that way, the protein is changing during development, and this is not something that's in the DNA in the gene. So this is only in the RNA. So it can change, and as you can see, it's changed during development. But it can also change when you activate the cell, and then this is going down. So you get back to the original state when you activate the neuron.
Let's see if I have this. So what I'm understanding, with my very limited understanding of this topic, is that basically the genetic code isn't just a set of instructions. Genes do different things at different times. So
yes.
we're looking at an enzyme that controls that sometimes it will do certain things and then turning things on and off.
Yes,
very simply put.
Yes. Because the DNA code is just dead. It's just study. It's what you're doing with it that is the interesting thing, and the expression from the DNA and the modulation of what's coming out of gene expression. That's where the brain is tremendous in variation here. We need a lot of different variations in order to.
So what are we learning from this knowledge? What does this knowledge tell us?
Yeah. So I'm just a little piece in the puzzle, and so we're coming from very small angel on this enzyme, this particular enzyme that is changing a lot of different receptors that are involved in neurotransmission, and this is something that is essential to have these changes.
So if the enzyme would not be working properly, what would happen then?
Yeah. So that has been done when you knock out this enzyme. So and the mice, they live for 3 weeks. They get epileptic seizures and they die, or they're not even born. So it's something that we need. It's not a mutation. It's something that we need in order to make
a building block. Or
yeah,
just like nutrients or anything
yes,
like that.
Yeah.
We need them to have a full set of all of these other things.
Yeah,
that make
to have the plasticity,
our bodies develop as they should.
The possibility to change. Yeah.
And I realized from when you're saying this, you mentioned epileptic seizures. Of course, maybe I should have said this at the beginning, but when we're talking about the brain, of course, it sounds like it's an abstract field of research, but we're talking in a very practical sense of of everything the human body does as well. So, for instance, when we think of mental health issues, which are culturally understood to be somehow maladies of the self, are of course brain illnesses to a very long degree, but also more physical things like everything from eating disorders and dyslexia to migraines and MS. All of these things are connected to the brain. So on a very practical level, getting more information about the brain it's more practically useful than perhaps space travel, even though it's similarly exciting. Yeah. Håkan, Fischer, you approach this topic of the brain and of understanding the brain from the very opposite direction.
Yes.
Coming from psychology, you start with human behavior, and then you try to figure out what the brain is doing. Is it how the brain is causing the behavior, or what the behavior is causing in the brain?
It's really how they are linked, so to say. So, as for example, if we are looking at some kind of memory function, we are looking in the brain to see where is the neural basis for this function, and then we are also interested in see how this function change. As for example, when we grow older, when we get better at something, and so on and so forth. So we are really looking at how they are linked back and forth, so to say.
So how would you go about studying the brain? What are some of the methods that you use?
Right. So we are typically looking at the brain on a large-scale level. That is, we are looking at huge systems in the brain, not at cell level. So we are really looking at both function and structure in the brain. So in terms of function, we are looking at, as for example, working memory function. Where is it located in the brain, function-wise? And then when we know where things are happening in the brain, we use other methods to try to see what is happening there. So both what and where are questions that we want to ask. And we do that by combining methods, as for example, fMRI with PET, which are two different methods. So
scanners, essentially, of different kinds. Yeah.
Different scanners. So by using multiple method approaches, we try to look at, as for example, working memory or emotional processing on different levels, and then trying to link these things together, both, as I said, in terms of function and structure. And also we use different methods for looking at function, because some methods are very good at finding out where in the brain something is happening, and others are good at looking at what is the temporal path, what is happening first and last and second, and so on and so forth. So by combining these things, we are trying to get a deeper understanding about the psychological mechanisms that we are studying.
Already I'm noticing that I'm finding it quite difficult to remember that when you say memories and emotions, I feel that in my body, I know what that is. It's very difficult for me to think of that as a chemical process, for instance, or an electrical process. What kinds of things can you learn? What are some examples of the kinds of things you're looking at with this method?
Right. As for example, we have done studies on working memory function, where we first target where in the brain things are happening, and then we are looking at what is happening there in terms of dopamine function. We are linking these things together, and then we give patients medicine that lower their dopamine level in the brain, and see what is happening both in terms of behavior, and that is that they're getting worse in terms of working memory, but also what's happening in the brain as a function of this manipulation. So that's one way of sort of linking all these methods and finding out not only where things are happening, but also what is happening.
I have some follow-up questions. I'm realizing on the dangers of trying that on actual brains, but I also realize that this is a good time to bring in our Skype guest. Joining us now via Skype is Dr. Bolte Taylor, who is a neuroanatomist, a best-selling author, and a public speaker. Her story of how she got a stroke and what she learned from this experience, both as a human being and as a brain researcher, has touched millions of people. Welcome to Crosstalks.
Thank you. It's great to be here.
Dr. Taylor, in 1996 you experienced a severe hemorrhage in the left hemisphere of your brain. You've been able to make a full recovery. How did you do that?
You know, on the morning of the hemorrhage, in the course of 4 hours, I watched my own brain deteriorate, essentially circuit by circuit. And I couldn't speak, or I couldn't walk, or I couldn't stay connected to normal reality. And the rebuilding process was the same. I had to approach my brain as circuitry and realize that every ability I had because I had cells that performed that function. And then it was a matter of figuring out how to get those circuits to come back online to be functional again.
Do you think that your knowledge of the brain helped you design your own recovery in a better manner?
Absolutely. Because I was a neuroanatomist and I look at the brain as though it is a collection of cells, it was a matter of which cells are working, which cells are not working, what is the relationship between different groups of cells, and how could I influence that in any kind of a positive way. So I really do believe that it was because of my knowledge of the brain that I was able to rebuild that circuitry.
You then wrote this book. It's called My Stroke of Insight, a brain scientist's personal journey about this recovery. What was the most important thing you wanted to get across in the book?
Well, I think the most important thing is that we experience neuroplasticity. When we look at the brain, up to really the last 10 to 15 years, we looked at the brain as this stable thing, and if there was trauma or damage, then we were going to have to live with that. There was no ability of the brain to recover. But in the last 15 years, we've learned that we do grow some new neurons. Neurogenesis is real, especially in response to trauma. We have neuroplasticity, the ability of cells to rearrange who they're communicating with so that we can actually learn and relearn things, or other cells can take over function. So I think for me the most important thing is that we are capable of recovery, and when we treat someone as though they will recover more than if we treat them as though they will not.
So I know that you're teaching people now that we have more say about what's going on in our brains than was previously thought. Is that what you mean by this? Let's see, did we lose you? Let's see, I think we have used it. Well, I'm sure Dr. Taylor will be back. Pardon me. Yeah, well, let's hope we can get Dr. Taylor back. It may be possible. I think, Håkan, you've likened the brain to a city when trying to explain the challenges of understanding the brain. Can you tell us a little bit about this analogy?
Right. Yes, I'm thinking about the brain as really a place for communication, right? So there's lots of communication on different levels in the brain, as it is in a city. You know, in a city you have different levels also of communication. You have big roads, you have underground, you have people connecting at different levels and doing it in different ways. So. And also, I'm thinking, because I'm interested in development questions also in relation to the brain, then I think that cities develop and grow bigger, and then they maybe get some problems in the cities with the roads and so on and so forth, as it is in the human brain when we get older, right? Things get sort of not really working as they did when we were younger. And to me, I think a city sort of picture is very sort of useful when you think about the complexity of the brain, because as it is in a city, a big city somewhere, it is in the brain, different levels of communication and everything at the same time.
So we would have little humans, and we would have districts, and we would have traffic systems, and all this. I was thinking about this image because of this idea. When Dr. Taylor was saying, well, here are this area. It's almost like this part of town was broken. Mm. We had to do some urban planning on the brain, essentially.
Right.
Let's come back to you, Jill. How can we train ourselves to control how we utilize and listen to our two brain hemispheres at different times or situations?
I think it's a matter of paying attention to what's going on inside of our head instead of just assuming that my brain is me, and so it's in control all the time. We actually have the ability to observe what's going on and pay attention to what's going on. And if we want more of that, then to continue running that circuitry and thinking those thoughts. And if we want to shift what's going on inside of our brain, then pay attention to that and give our brains other things to think about. We are very malleable, and mindfulness, the ability to pay attention to what our thoughts are, that it's circuitry that we're running, and to be able to change our thoughts and actually change the biology and the anatomy underlying that. We are at a phenomenal time in being able to really pick and choose who and how we want to be in the world.
Well, do you agree? Does it feel natural to you others to think of the brain also in this way, some ways as self-programmable, that you do create your mind? Yeah, Marie?
Oh,
Yeah, I think definitely that's something that you can see also on the cellular level, when like the nerve cell, when you think and you use the nerve cell, you will increase the dendrite outgrowth, and so you will, the memory and learning will develop when you think. So definitely I think you have an extreme plasticity and possibility to change your own brain. The more you use it, the more developed it is.
So between what you're all saying, it sounds like we are at a moment in history when self-help books will maybe not become a thing of the past, but certainly they might be based on actual science and not just random snippets of science, as they often are now, where you can say, well, actually, this kind of meditation practice will make your brain healthier, or this kind of, whatever it is, this kind of pedagogy in schools will actually make us smarter. And so do you think that's a reasonable prediction for the next decade or so, Anders?
Yeah, I think, you can really do a lot. And in my special field, we talked about memory. So if we think about memory and how memory is dependent on that we are interested, for instance. This has to do with the modulatory systems in the brain, the dopamine systems. So we can really think about that. If we are interested in a subject, we will learn about it, and we will maybe dream about it, and we can sort of control ourselves. If we are not interested, then maybe it's not the point to study at that time. So there are many actually connections between how the computational models of memory work, and they can be connected to actually experience of improving our memory capacity. If we sometimes say we want to remember something, we should connect it to something interesting, to something funny, and we should connect it in a bigger context. And this you can understand that from the network models, that the bigger you can engage a network, the easier it will be by this plasticity to actually be active and make a difference.
There's this medieval practice called memory palaces, this idea that you build sort of pictures of buildings in your mind, and then you place your knowledge in different places, in different rooms. And but you're saying that scientifically this works?
Yeah, even the models that we think about and develop, it is a natural effect of them. You can understand why this is effective from these models. We haven't discovered this from the models, but we can at least somehow explain it in a way.
Marie, your research shows that the molecular structure of the brain changes in reaction to external stimuli and also during a lifespan. This is exactly what we're talking about now, right?
Right. Yeah. Exactly.
So what are some of the things that are most important that affect the brain? And maybe suddenly I'm asking out of personal curiosity, but maybe there are some important scientific answers also. But what kinds of things affect us on the molecular level?
So I think you have to think of, on the molecular level, you have to think of regulation, like how genes are regulated, and how that is affected by stimuli, for example, because there's an extreme amount of regulation is happening during a stimulation process, both out in the synapse, but also in the whole cell, with transport of different molecules, both RNA, proteins, other molecules. So it's a lot of different regulation on a lot of different levels, and you have an extreme secure system to keep things going. I think so. So that is very important in order for a cell to function. I mean, any cell in the body, but especially in the brain.
I'm not entirely certain, actually. But you know, you said many things, but can you say what kind of things affect us most?
Take proteins.
Yes.
I mean, we have about 30,000 different genes, but we have more than 90,000 different protein variations that comes from these genes. So that means that every gene is giving rise to a lot of different proteins. So, we're using these different proteins in different ways during different times. And also, I mean, if you, during growth, when you're stressed, when you exercise, when you're happy, when you're sad, I think you can always think of molecules that are regulated.
This is exciting, and because I'm thinking, this is happening on so many levels at the same
Right,
time.
yeah.
I find it very difficult to zoom in and out. Does it all come naturally to you? Does it feel that is it simple to think of the brain on all of these levels at the same time for you? I'm just checking. No, not entirely
No.
simple. Oh, that's a
I guess
relief.
we're focused on different areas. I mean, my cell can also be a city, just like your brain is a city. My cell is a city with different things happening.
There's something interesting here. Jill Bolte Taylor, do you feel that your experience of this stroke, did it change the way you do science? Did it change the way you view the brain yourself?
It did. It gave me, well, first of all, in the absence of my left brain, in the absence of language, everything changes because I was still conscious, I was still thinking, I was just looking at everything in the bigger picture, and the details were not what was important to me. So a lot of my philosophical or spiritual understanding of circuitry completely shifted. And my perception of circuitry and the ability to bring it online or take it offline or to invite it back online or to leave it no longer going on, everything changed in how I could actually look at my city and build it from scratch and determine what did I want, what did I not want in that city. And it did give me a completely different perspective of the brain.
That's interesting. Håkan, do you have something on that?
No. I totally agree that it must have been very, very interesting experience to sort of have this dual perspective of both know so much about the brain, but then also experience this change sort of simultaneously. It must really have given a much deeper understanding of the brain than if you just have it on a theoretical level, of course.
I know that there is, I mean, this may sound incredibly banal by comparison, of course, to this life trauma that we're also talking about here, but there are experimental methods where you can turn off parts of brain functionality. I'm looking at you a little worried, yes. Håkan, because you told us about this. So can you just explain how we do that and why it's not very wrong?
Right. Yeah. So there are now methods that enables us to sort of shut off different parts of the cortical areas of the brain for just seconds or maybe like a couple of minutes or so. And these methods are called TMS, and there are different types of this, where you put a magnetic coil on top of, as for example, the language area or the frontal lobe somewhere, and then you sort of by making a magnetic field around this coil, you can sort of turn off the electricity which cells use to talk with each other in an area, and then you can see what is happening when that area doesn't work as it should. As for example, if you're looking at brain function related to language, you can put it over the language area and take out the speech function or the language comprehension function and so on and so forth. So this is in a fairly new method that makes it possible to really target down what is happening and not only where it's happening.
It does sound quite terrifying. Is this something that it's entirely safe, I would assume?
Yeah, it's safe as far as science knows today, yes.
As far as science knows today.
As many things are, right?
I guess that's life. That's life for you, right there. I don't know. Has any of you three ever had parts of your brain shut down?
I have.
Well, what was that like?
It was a very surreal feeling, I must say, because I was talking, and then this cord was put over my Broca's area on the left side, and it was turned on, and then I was talking, and suddenly I didn't talk anymore. And then it came back again after it was shut off. So that's one example of how you can experimentally sort of look at causality between, in this case, language and an area of activation in the brain, which is very difficult to do if you don't have these types of methods, because otherwise you can just see where things happen, but you are not really sure that it's really this thing that we're looking at that is what we're also looking at in terms of behavior.
So at the risk of sounding, again, incredibly unscientific, which I am, I wonder how much of this becomes, on this level, when we're talking about what you are able to do and how you think about things, and so this becomes also the study of personality and identity on some level. It's very difficult not to end up in a very profound area. I don't know, Dr. Taylor, would you agree? Were you a different person when your brain didn't function the same way?
It absolutely did. The two hemispheres are very different in the way they process information. They're processing the same information, but very differently. And so the right brain is looking at the context and the bigger picture and the relationship and the similarity. And that's going to have a value structure that is going to look very different from the left brain, which is looking at details and individual. So the two value structures of the two very different characters, for me, were extremely different from one another. And as the left brain character went offline, the right hemisphere character came back on, and she was very different. So I'm very different now than I was before. And I had the option of returning to the value structure of my left brain as I regained those abilities, but I actually preferred the value structure of the right brain, so I am very different now than I was before.
It sounds almost like a religious experience.
No, actually, from my perspective, it was not, but I don't use that language. If you look at the brain and the circuitry underlying religious belief, that's going to be something that's going on in the left brain. In the absence of the dogma and the story that we tell ourselves and one another about religion, the ultimate goal, of course, for virtually every religion that I'm aware of, is to have that right brain experience where we are connected. And you can use that language, but for me, I'm more comfortable in the language of the right brain and the left brain, and the ability, any ability we have because we have cells that perform that function. So my ability to have what someone else would call a spiritual experience, to me, is the circuitry of the right brain.
That's fascinating. And I think in some ways this brings us back to the very beginning, which is how on earth do you model all of this? And we are all of us have, again, like my brain is shutting down at this information in some way, because I have all of these processes are going on automatically all the time, and unless I put a coil on my head, I can't shut them off, or possibly through some meditational practice and so on. I'm sorry, I'm going to bring it back to you, Anders. How do we model this in a meaningful manner?
Yeah, I mean, we have to conquer this big problem by dividing it into pieces. So obviously you don't start with the whole brain. What we actually do is starting by building models of individual nerve cells, neurons. There are lots and lots of data about neurons and different types of neurons. So if we want to build a model of cortex, we can get that data from published sources and build neuron models. Then we can look at the synapses, the contacts between them, the transmission of signals, and do the same and mathematically formulate that. And then we do the plasticity, for instance. So we have these, and we can build a network by replicating these components. And then we have a small neural networks like the cell culture that you had. Actually, it's a good idea to model these kind of things. But of course, that's not the entire brain. And then we build large networks of that, study the memory. We do virtual brain experiments in the supercomputer and compare how this neural network dynamics then that we can achieve, how that compares to real measured brain activity, you can say. And then we can evaluate the model based on this.
Can you say how much, you mentioned a supercomputer. I'm realizing now that actually one would need quite a lot of processing power.
Yes.
The reason we don't build a full copy of the brain, I'm understanding, is we have no idea how it works. We have some idea how it works. But is there also just a capacity issue?
I would say today, actually, 10 years ago, I was really discontent with the computers we have. But now that from technological development, this barrier at least is removed, actually. So it's not a capacity problem.
Quite recently, essentially, it has been.
Sorry?
So it has been removed relatively recently.
Yes, I would say it has. And so now we can build more complex models than we can sort of handle, let's say. So we can now simulate the type of models we would like to study in a reasonable time with turnaround times of minutes or something like that. So that is not a real problem. It's the lack of data for building the models, for instance. I mean, we have tremendous amounts of data from experiments. The only problem, or the slight problem, is that when you build a model and start to ask about these specific data points that you want and you look for them, they are not there because they are very hard to measure probably, and the instruments are not there yet. So this makes it hard, of course.
So there's a gap between what we know should be there from the models, and what the experiments can actually measure.
No, well, we need data to build the models in the first place. For instance, the learning rule, the synaptic plasticity rules, we need to express them mathematically in a quantitative manner, and measurements cannot tell us that yet. So we have to make guesses about and fill in these gaps. And then we have our model, which is built partly on guesswork, and we simulate that and evaluate it. So it's more that we have a tremendous amount of data, but not exactly the data we want.
Okay, so if we look at that again from the completely different perspective, Maria, so what you're doing is basic research. You're approaching generating parts of this data that is needed. So what is satisfying to you about that? And do you see a point where it connects to practical applications, or is that quite far away?
No, I know. To a certain extent, I mean, of course, you can in the long run you can see connection to be able to explain different diseases like neurological diseases, for example. But that's not really my focus. My focus is to understand what is going on. I think it's super exciting to just try to understand how things are connected and how flexible the cell is. But of course, I mean, the more you know about this, what is happening on the molecular level, the more you can understand what is going on during a disease and when something goes wrong, and then instead of treating the disease with something that you're just guessing, you can make a good guess by in detail fix that problem, and that's something that we're close to be able to do now. We can fix.
So medicines, for instance, just again a trivial example, medicines will become much more precise.
Yes, yeah, because side effects, they are usually because they are too crude. The chemicals are too crude. Because they are firing on a lot of different things that you shouldn't be at. So the more precise you are, the less side effects you have.
So I'm curious now, for the other three, the kinds of computer models that Anders is working on, what could they bring to your research? Do you think they could? You're looking like, of course it could.
No, yeah, I think
If we had, well, maybe I should ask you, Anders. What do you think? What could your tools or other types of ICT tools be able to offer, for instance, to the kinds of research that Marie is doing?
Yeah, I mean, first of all, I'm sure Marie is using lots and lots of IC tools for doing her research, instruments and things like that. But in terms of modeling the dynamical biology of the cell, I mean, this is, I mean, it is related, but it's maybe a little bit different from you're doing. You're looking into the, as I understand it, the components of the cells at a certain instance, the expression of proteins. So you have the protein-based machine there, and not really going further and looking at how that functions in dynamically. Maybe I don't know.
We're getting there.
If you're getting there, that's I think if you
So
can sort of provide us with a machine and the biochemical pathways, and ideally the parameters, the exact numbers of how these reactions occur, which is very hard. Then you can start to model the biochemical processes inside the cell. And actually then you can also hopefully see what data you're lacking. What is it that this model is very uncertain in certain aspects? And then we go back to Marie and ask, could we somehow measure this thing? And in that way you would work together around such a model and develop it. And I think that model eventually will also help in the drug development even, because you can challenge it with different chemicals in simulation and understand what happens. So it is really something that can be very interactive.
It sounds like the research is approaching each other, so that very soon, no, not very soon, you made a face there, relatively soon. What does soon mean in this field? You can, are we talking about within 3 years or within 30 years?
Yeah, that's of course very hard to say, but So, but there are examples actually of such similar things. We have looked into a little bit the beta cell and insulin secretion, important in diabetes and stuff like that. And there are actually drug companies investing in modeling, dynamical modeling of this system. So it is in certain islands going on.
Very good.
Very
good. I would like to take a question from the audience if someone has one on their mind. Yes, please. And there, you need to move down to the thing, and you as well. Let's see who makes it first. That person gets to ask first. And turn it into a little game. There we are. Yes, all right. You go first. Please state your name and your academic affiliation, please. Yes. Yes.
Hi, my name is Deepta, and I study at SU. And my question was that in terms of ADAR that can make these kind of changes in the transcription of DNA, it seems to be creating a flexible system in response to stimulus. Have you ever seen ADAR or any other enzyme system acting differently in different brain cell populations in response to different stimulus?
Oh, that's interesting.
So the ADAR is this enzyme. I didn't think I mentioned that, but that is the enzyme that is making this change. And so far, we haven't really been looking at different parts of the brain. We're looking in the forebrain, and that's where we've been analyzing. But if you look at some of these editing events, so it's called editing when it's changing the RNA, it seems like it's uniformal. Most of them are uniform. Like in the GABA receptor, for example, there's an editing event, and that is happening. We have looked at different parts of brain tissues, and it's highly edited. Up to 90% of the RNA is edited in all different parts of the brain. So, but of course you can imagine that you can have a change there, and you can have a change within the cell even. So different parts of a cell have different editing events.
But this kind of flexibility, what does it mean computationally? Can you create this kind of flexibility in a chip, essentially a chip that can evolve over time, make changes to itself? Is that even possible?
I guess we have to look at Anders.
Oh.
What is it, computational? What's the computer version of this?
Yeah. Well, that kind of chip, it's certainly possible to model plasticity and changes on a chip. But if this particular system is suitable for that, I mean, we are thinking in those terms, but then it's the networks of the brain, the synapses, and how they change. And I'm sure it's much less complex than what Marie is telling us, but at least there are changes happening. Yeah, and in relating to learning, for instance, this is already been done on chips. So yeah.
That's exciting. Thank you very much for your question. Thank you, Deepa. And the next question, please.
Hello.
Hello.
My name is Jacob, and I'm a student at Grilska Gymnasium, or how you want to say. And I was a bit late, so this may be a relevant question or a dumb one. But I was wondering for the memory expert, if, for example, in shows like Continuum, there is a medicine that makes you be able to remember happy moments in your life. Do you think we are in a grasp of that, and if so, when?
Okay, I'm going to broaden that question out a little bit. Thank you for your question, Jacob. So indeed, in many popular culture settings and in popular understanding, there is this idea that all of your memories are there. And everything you've ever experienced is in your brain if you could just retrieve it. And now if everything you've said is all right, we should be able to figure out a way to retrieve them. Is this possible or not?
I think, first of all, one has to realize that memories are very subjective. They change all the time. As, for example, they change every time that you remember something. It changes because it sort of becomes re-encoded again every time that you sort of again remember your favorite girl or boy or whatever. This memory will be changed. So memories are flexible, and they are not there forever in your brain. That's my take on this.
So the more I remember, the more times I think of something, the more time, the further away from the truth or from the original experience it is.
Yeah.
That's depressing. So I think that would be a no to your question. Yes.
I think there is. I think it would not be very good if we remembered everything we experienced, and I think rather that memory is rather selective, and that there is evidence of that in terms of memory consolidation. What actually, from your experience during a day, what makes it into your long-term memory are things that are interesting, that engage you, that where you were upset, frightened, or happy. Yes. I mean, so, okay, remembering only the happy events, I'm not sure one can be that selective, but I think memory is very selective in remembering the important events. And I think that's rather lucky, because otherwise, because memories can also interfere with each other. So if the unimportant memories sort of interfered with the important ones, it would sort of mess up the system, I'm afraid.
Thank you for your question.
Can I ask a follow-up question?
No, thank you. We're running out of time, unfortunately, and I have to ask the very important question, since we are addressing a global audience of young researchers. If you're thinking about going into the brain, so to speak, in some way, if all of you were now, let's say, in your early 20s, what is the most exciting field from all of your perspectives? Where would you like to go and study right now? And we will just do a very brief round. Dr. Jill Bolte Taylor, what would you go? What's the frontier?
I would go into neuroplasticity and the ability to pick and choose who and how we want to be in the world. I think that becoming a more mindful, conscious humanity will change the planet.
It's wonderful. Anders.
Yeah, I will be boring, because I will say I would try to become exactly what I am. I would study computer science because I love computers, and I would also use them to study the brain, because that's an extremely fascinating and challenging subject. It is challenging also because you have to learn much, not only about computers, but about biology and neurobiology. So, but that would be my track.
Very good. Marie?
Yes, I think I agree with Jill that the neuroplasticity is something that is extremely exciting, and of course I think, reflecting to my own research, also do it on the cellular level, on the neurological level, where you look at the plasticity, what is happening during different external stimuli. I mean, I think that's extremely important. And interesting.
And finally, Håkan,
Yes.
where's your frontier?
Yeah, I'd love to go into the social brain, because in my view of the brain, it's wired to be social. So I would like to go into the social brain and find out what it does and why it does what it does. And then in the long run, I would like to look at this from not only in one brain, but in many brains that are communicating. So that's my future perspective, that it will be so interesting to look at the social brains, that is, brains in a group, and how they interact, and how that change as a function of development also.
How we choose who we are and
Yes.
how we shape each other. That's wonderful.
Yes.
Thank you so much. We could talk about this for hours. Unfortunately, we are so out of time. Thank you so much, Marie Öhman, Anders Lansden, Håkan Fischer, and Jill Bolte Taylor for joining us tonight. Crosstalks will be back in a month with more great minds and intelligent discussions. Be sure to check in at crosstalks.tv for updates on topics and on guests. But for now, be safe and be brave.